Welcome to our today's episode, which is the fourth episode based on biology questions and answer series. In fact, we're going to begin with the first question on this list, question 31, which of course you can see other episodes from the link below this video. So this is the fourth episode of this series. Let's begin with the first question here, which is which nitrogenous base or bases pair together in DNA? Which nitrogenous bases? So we are looking at A option, AG and CT, B option, AT and CG, C option, AU and CG, and the last option, GT and AC. Which among this list is the correct answer? I give you a few seconds to look through it. So looking at this option, the correct option here is B, which is AT and CG. That's the exact complementary strand when it comes to pairing of the nitrogenous bases. And this, of course, you need to keep it as a formula at CG, where A and T and C and G. This is the correct answer. Of course, at the end of this, make sure that you comment below this video to tell us exactly, out of this top 10 question we're going to be sharing today, tell us how many correct answer you had in the list of these top 10. Make sure that you are very honest in the comment section to tell us how many of these 10 you have correct. So let's move to the next question here. Let's move to the next question. Question number 32. Which molecule is the primary energy currency of the cell? A, we have ADP, B, AMP, C, ATP, and D, NADH. Which here is correct? The correct option here is C, which is ATP, because ATP is the primary source of energy which is used for cellular processes. So the correct answer is ATP, which is the energy source. Let's move to the next question. Question number 33. Please make sure that in the comment section of this video, you comment to tell us how many correct options you have out of the list of 10. Be honest and leave the comment below this video. Let's proceed to question 33. Which organelle modifies and packages protein? A, ribosomes, B, endoplasmic reticulum, C, Golgi apparatus, and D, lysosomes. Which is correct in this option? In fact, the correct option is C, which is Golgi apparatus. Golgi apparatus is or play the primary role of processes of protein. That is process protein for secretion and transport within the cell. This is the option of the Golgi apparatus. So the correct option is C, as you have seen here. Let's move to the next question. The next question, which is question 34, we are looking at which metabolic pathway occurs in the mitochondrial matrix. A, glycolysis, B, Krebs cycle, C, Calvin cycle, and D, fermentation. Which is correct among this option? The correct option is option B, which is the Krebs cycle. In fact, the Krebs cycle is the one that completes glucose oxidation. It functions in the completion of glucose oxidation, thereby producing electron carrier for ATP synthesis. So that is the major option of what it happens. So the correct option is the Krebs cycle in this option. Let's move to the next. Let's move to the next option, which is option 35. Which molecule accepts electrons at the end of the electron transport chain? Which molecule accepts electron at the end of the electron transport chain? We have option A, which is carbon dioxide, option B, oxygen, option C, NAD+, and D, C, NAD+, and D, water. Which is correct in this list of options? In fact, if you looked at it, the correct option here is option B, which is oxygen. In fact, oxygen acts as the final electron acceptor after what we call, that is, for the molecules to accept electrons at the end of the electron transport chain, it is the oxygen that acts as the final acceptor of electrons. And when this happens, it tends to form water and, of course, allowing ATP production to continue. This is how the process occurs in this chain. First, the oxygen accepts the electrons as the final acceptor. When this happens, it tends to the formation, it causes water formation. And once this occurs, it processes or proceeds to the production of ATP for the process to continue. So let's proceed to the next question. The next question here will be option 36. Which type of mutation does not change the amino acid sequence? Which type of mutation does not change the amino acid sequence? We have A, we have missense mutation, B, nonsense mutation, C, silence mutation, and D, Frameshift mutation. In fact, if you look at this option, the correct one here is going to be C, which is silent mutation. It is the silent mutation that does not change the amino acid sequence. Why? Because the silent mutation can only change DNA bases, but without altering the encoded amino acid due to genetic code redundancy. This is the reason. It can only affect the DNA base itself, but it cannot change the encoded amino acid itself, which of course it is due to as a result of genetic code redundancy. This is why option C, which is silent mutation, is correct. But just keep in your head that anytime they talk about silent mutation, it means that it does not alter the amino acid sequence, but it can only change the DNA base, but the sequence of the amino acid cannot be altered. This is why you need to know that the correct option is the C, which is silent mutation. Let's move to the next. Let's move to question 37. Which process converts RNA into protein? Which process converts RNA into protein? We have A, replication. We have B, transcription. We have C, translation. And we have D, translocation. Which here is correct? In fact, if you look at it very critically, you are going to note that the C option is the correct one, which is translation. Because, of course, translation occurs at ribosomes. And, of course, this is where the mRNA codons are decoded. And, of course, when they are decoded, they are changed into amino acid sequences. This is why it is correct, because the process to convert RNA into protein itself. That is why it is only occur during translation. That is why translation is going to be the correct option, because it occurs at ribosomes where messenger or mRNA codons are decoded into amino acid sequence. So the correct option is C, which is translation. Let's move to the next. The next will be question number 38. Which factor increases enzymes activity up to an optimal point? Which factor increases enzyme activity up to an optimal point? We have A, extreme pH. We have B, high temperature. We have C, increasing substrate concentration. And D, enzyme denaturing or denaturation. Of course, if you look at it very critically, when we talk about the factor that increase enzyme activity up to an optimal level, you need to understand that it is when the substrate concentration has been increased. In fact, if you increase the pH, temperature, and enzyme denaturing, nothing is going to happen in terms of the increase in enzyme activity. Enzyme activity can only increase when the substrate concentration have also been increased. Of course, this is as the result of knowing that the more the substrate increase, enzyme substrate collision will also increase. And of course, you need to understand that when the substrate concentration is being increased, enzyme activity will also increase very automatically or rapidly until saturation point is met or reached. That is why increase in substrate concentration result to increase in enzyme activity. I believe you know about the lock and key hypothesis. If the substrate concentration is increased, there's also going to be more increase in enzyme activity until when the saturation point has been reached. That is when it will score. That is when we know about the optimum point. So correct option is C, increase in substrate concentration. And of course, C is correct. Let's look at question number 39. Let's look at question number 39, which is which type of RNA brings amino acid to the ribosomes? Which type of RNA bring amino acid to ribosomes? A, we have mRNA. B, RNA. C, tRNA. And D, S-mRNA. Which of these RNA brings amino acid to ribosomes? If you look at it, the correct answer is option C, which is tRNA. In fact, T means transfer. Transfer RNA act as carriers. That is, the carry specific amino acids corresponding to the mRNA codons during translation. So they act as transport, which of course carrier transport, which the T simply means the transfer of it, which is tRNA. So they result in the carries or the carrying of this amino acid corresponding to the mRNA during translation. That is why it is tRNA, which is option C. Let's proceed to the next question 40. Question 40, which carbohydrate is used for energy storage in plant? Which carbohydrate is used for energy storage in plant? We have A, glycogen. We have B, cellulose. We have C, starch. We have D, chitin or chitin. Which is correct? In fact, the correct answer here is C, which is starch, because plant store glucose as starch, and therefore it can be easily broken down for energy when it is needed. When energy is needed, the starch will be broken down for the energy release. We cannot say glycogen. This is in humans. We cannot say cellulose. Of course, we cannot say chitin, and the correct answer is starch, because energy, that is glucose, is stored in the form of starch, which is, it is being broken down to release energy whenever it is needed. That is why option C, starch, is the correct answer in terms of which carbohydrate that is being used for storage that is which carbohydrate is used for energy storage in plant. Of course, if you want to see more of these upcoming series, this is the 4th episode. We are going to release the 5th very soon. If you want to keep an update on the series, we keep updating on the best top questions and answers with some reasons why those answers are correct, so that you stay ahead of others to keep track on the most and best questions you can ever find when it concerns biology. And of course, you want to make sure to go ahead to like, comment, and subscribe, and also enable the bell icon notification, so that anytime we release another new video tutorial like this to answer some of these questions, then of course you are going to be the first to get notified and see what update we have for you. So what you need to do is just to make sure that you just subscribe and enable the notification bell icon, and possibly also like the video to tell the algorithm that you actually enjoy the video. So see you again in the next upload, and bye-bye for now.